Welcome to Chapter 10, a big chapter, one of the bigger chapters in this unit. We will be dealing with somatic senses in the beginning, and then we will finish off with special senses on the backside of this series of lectures. I fully expect at least 3 or 4 segments for Chapter 10. The reason why I dedicate so much time to this chapter is the fact that it is so directly relevant to signs and symptoms. And given the population of this class is often with physical therapy and occupational therapy. So we're going to go through some general properties of sensory systems, focusing at first on the somatic senses, which is like things like proprioception and on the surface of your body or your body position and that sort of a thing. Then we'll get into the special senses. We'll talk a little bit about smell and taste. We'll do hearing, a little bit of balance. Hearing and equilibrium are in the same family of senses. They both use hair cells. And then lastly, our mechanism, the one that we are breaking out the mechanism, is the eye and vision. We're also going to do a little pathophysiology and physics here with nearsightedness and farsightedness and astigmatism, because again, that's such a major part of people's lives. Vision is one of the most important senses to function in a modern world, so we're going to spend a little more time there. But understanding senses is to understand perception as well. It's not only about what the stimulus is, but what the stimulus is in context. We don't have time to do all of the contextual stuff. That's certainly an important aspect of psychology, brain and behavior type classes. Nevertheless, we're going to work on what we call discrimination. That is to say, is it touch? Is it sound? Is it near, far, or where is it? We call that discrimination. Has a nasty set of connotations with it, but in physiology terms, it is an exceptionally important one. So what separates what we call the somatic senses from the special senses? Well, the number one criteria has to do with the fact that somatic senses are almost always modified nerve endings. Yes, they're differentiated. Yes, they're highly specialized, but nevertheless, they do arise from neural cells. Whereas the special senses arise from a specialized receptor cell. So the receptors on the somatic senses are nerve endings. The special senses that you have a specialized cell that then feeds into a neuron. And whether we're talking about somatic stimuli, which includes the level of length and tension, or the position in 3 dimensions that we call proprioception, or the visceral stimuli, all of these things we process subconsciously. It may rise to the level of central nervous system attention, like for instance, if your blood glucose drops, you might begin to get hungry. If your osmolarity drops, you may begin to get thirsty and so forth. But nevertheless, it's processed at the subconscious level. And then if it rises to a certain threshold, then the higher order neurons are brought in. So sensory pathways, this signal and response pathway is fundamentally signal transduction. Just like we talked about in Chapter 6, talked about a little bit in Chapter 8, and so forth. What we begin with, though, the input is a physical energy, either the presence of something like heat in this case, or the presence or absence of a chemical ligand, or physical pressure, any of those things that would stimulate a receptor to then, and the receptor in this case is a neuron, and it's turning that initial signal into first graded potentials. First a graded potential, call that a receptor potential, but it's a graded potential. That graded potential, if it reaches threshold, then becomes a set of 1 or more, probably a lot more, action potentials to the central nervous system. Once it reaches the central nervous system, we integrate it with the appropriate effectors, depending on intensity, depending on the mode of the stimulus. How intense is it? Where is it coming from? Is it something that should be acted on immediately, or is it something that requires a deeper, more psychological response? Here are some examples of those neurons again, but these are the examples of the receptor neurons for somatic senses. Free nerve endings are the most generic of the receptors. These guys are sensitive to a whole lot of things, but they have what we would call a higher threshold. That is to say, we're talking about pain here. So if the stimulus is intense enough, even if it's multiple modes, like maybe light or heat or chemical presence, etc., these guys respond, and oftentimes that perception is pain. Then we have other ones that are more complicated, like the Pacinian corpuscle here. It's a form of touch receptor. I'm not going to have you break out all of the different kinds of receptors, not anatomically anyway, but understand that this is still a free nerve ending. This one just happens to be myelinated, okay, and so forth. But again, you have a stimulus that then triggers graded potentials in this area, which then can lead to action potentials sent to the central nervous system. And over here on the right, here we have our special sense. And this is a specialized receptor cell. This happens to be a hair cell in this case. What happens is we get a stimulus, causes a membrane depolarization event of some kind, and then we secrete neurotransmitter across a synapse to another sensory neuron. So somatic over here on the left, special over. Very important concept right here. We've dealt with this already. We talked about different receptors in the first exam, and this is where I kind of noted that thermoreceptors, are they mechanoreceptors, or are they really thermoreceptors? And I tend to skew to this interpretation that thermoreceptors are, while they may have a mechanoreceptor type in a cell, they are in fact sensitive to heat. Certainly in our macro senses, these guys are sensitive to heat more than they are physical pressure. Okay, it's not to say that they wouldn't feel like heat if you had certain types of pressure, but we're going to break that out separately this time. So we have chemoreceptors that are receptive to different chemicals, like a ligand-gated channel kind of a thing. We have mechanoreceptors that are for cell stretch, osmoreceptors, vibration, acceleration, sound. These are all what we call mechanoreceptors. Photoreceptors, photons of light, and then thermoreceptors, varying degrees of heat. Now, here is the key concept. Here is the key concept. The type of mode that the receptor is most sensitive to, we call the adequate stimulus. Students year after year after year, and gosh, I wish I was the king of physiology because I would change the name of this. Most people who aren't really focused will screw this up. They will mistake this for threshold. Okay, maybe a way of putting this is that it's the type of stimulus to which the threshold is lowest. Maybe that's the best way of putting it. In any case, the adequate stimulus is the mode of stimulus, whether it's chemical or mechanical, photo or thermo, that it is most sensitive to. All that said in that previous slide, and you should flag that one and draw that out and write down that concept. Get to know that concept. Stimuli can and do stimulate multiple receptors simultaneously. For instance, I was lucky enough to be able to see the space shuttle launch back in 2006. And at the time when it launched, we were miles away. We were 6 miles away from the launch site. And when it went off, it wasn't only sound that you heard, you could feel it in your chest. You could feel a physical pressure wave in your chest. And so while the mode may have been sound, it was of enough intensity and frequency that it triggered baroreceptors in my skin and probably in my lungs too. It was absolutely one of the most awesome things I've ever seen. This picture doesn't do it justice because those flames actually glow kind of an indigo purple that is just amazing. I had a similar experience even a few years earlier. I took this photo up at the fly-in in Oshkosh. They have this big, becomes the busiest airport in the world in Oshkosh, Wisconsin. And the Concorde was still flying then, and they were offering flights on the Concorde. You could go take an hour-long flight. This thing has afterburners. It's absolutely stunning when this thing takes off, and I was lucky enough to be on the flight line close to the runway, so we could be within maybe a couple hundred yards of the runway. And when this thing went off, it was that same sensation. The noise was enough that you could palpably feel it. And so modes can and do cross if they are of sufficient magnitude. But again, adequate stimulus gets at the idea of the stimulus to which the receptor is most sensitive. But remember that sensory transduction is signal transduction. We get a stimulus of whatever kind that creates a graded potential. That graded potential then creates action potentials sent up this afferent neuron to the central nervous system. It can be what we call an ionotropic receptor, a receptor channel, or we have other types of metabotropic receptors that initiate membrane potential change using second messengers. So maybe they create a second message, and then that triggers a ligand or ligand gated channel gated to an internal presence of second messenger that then causes a membrane change. I'll show you that figure here in just a second. But again, with adequate stimulus, the membrane potential change is going to be higher for a smaller amount of signal. That's what we mean here. The membrane change, the membrane potential change will be greater in magnitude for less energy from the stimulus to which it is most responsive, and that's the adequate stimulus. You can trigger, and you can, if you do this really, really carefully, you can actually, if you close your eye just ever so slightly, and then you press gently on the corner of your eye, if you look on the opposite side of your vision, you might see a little cone of light with a little blue. For me, that's what I see. I see a little kind of a bright halo around a blue air, kind of a bluish area. That's where the pressure is triggering photoreceptors. So it's physical pressure that's activating photoreceptors. So the photoreceptors are still most sensitive to light, but yet they will still be responsive to other stimuli. Just like our sound with the physical pressure in the previous examples, adequate stimulus. If we hit threshold, regardless of the stimulus, if we hit threshold, then we will send action potentials. But with adequate stimulus, we'll send a lot more action potentials with less energy than we would for other signals. It usually takes a lot more energy from the non-adequate stimulus to get action potentials. But that membrane potential we call receptor potential, but it's the same thing. It is a graded potential. It's just that the graded potential will be bigger with adequate stimulus than it will be with other stimuli. Free nerve endings are sensitive to all kinds of stimuli. It's just that their receptor potentials don't kick in until there's an intensity that may be directly possible to create injury, okay, or cell damage. That's what the threshold is for free nerve endings. And here's the figure that I was referring to from chapter 6. Over here on the left, we have an ionotropic receptor, and this one's gated for the presence or absence of a chemical, but we could also have it for some other stimulus. The adequate stimulus could be different for this one. Creates a change in membrane potential, or we could have a receptor like this one that creates a second message that then triggers that channel, or maybe the G protein does it directly. It's possible. In any case, we now have a change in permeability. We get an electrical signal. That's what we call a receptor potential. If it's of enough magnitude, the voltage sensitive protein, in this case, if it's a neuron, that would be the double gated, voltage gated sodium channel in the axon hillock, which then sends action potentials to the central nervous system. But it can be ionotropic this way, directly, or it can be metabotropic, either by the G protein or a second message subsequent to the receptor that then activates that channel. In addition to the cellular makeup of these neurons, we also have anatomical considerations that play a role in perception. Look at this figure right here. We have a receptor neuron, another receptor neuron, and a third receptor neuron. They're all here on the skin surface so you could feel it, but they all are synapsing with a single secondary neuron. So what does that mean? That means if we poke over here in the green, or we poke over here in the purple or in the yellow, we would feel it in the same place because there's only one secondary neuron. This is known as a receptor field. And we can actually do that experiment. I am working on a lab protocol that you guys can do to do this. It might require a partner. I hope you guys can give it a shot. With COVID, that makes it a little trickier. But if you are living with someone, or if you have someone in your family that can help you, or you could do it on them. That's possible too. If you have family that and so forth, observing COVID protocols. In any case, if you had this type of a situation where you're poking two of these fields simultaneously here, you would not be able to discriminate that as 2 points. You would feel it as one. And the receptor fields, depending on the area of skin that you're talking about, can be actually quite large, more than an inch in some cases. This is known as a receptor field. It's the primary sensory neurons and where they synapse into a secondary. The smaller the field, the smaller the finer the ability to do what we call 2-point discrimination. And so here's an example of where if we have fine touch areas, we may have a very low level of convergence. You can see that only one, maybe two receptors are synapsing into secondary neurons. And so 2-point discrimination in this area is actually quite small. In some cases, maybe only a couple of millimeters in some cases. Both receptor fields can be equally sensitive. They can both detect very, very fine touch. It's just that they can't locate it as clearly as you can in smaller receptor fields areas. Again, we're going to try to do this lab. I'm going to give it a shot. I'm going to cross your fingers we can do it. And you can measure these things. You can see that in some cases, the receptor fields are 60 or 70 millimeters. That's 6 or 7 centimeters. Take your fingers and spread them out a couple of inches. That's how big the 2-point discrimination is required. We'll give that a shot. We'll give that a try. Well, we have the architecture of the neurons. We have the subcellular stuff. What about perception? Gets close into psychology here. Again, how we take that information, send it to the central nervous system. How does the central nervous system interpret that information? Like, for instance, visual and so forth. We see this apple right side up, despite the fact that on the surface of your eye, that apple is actually upside down. Pretty interesting stuff. A lot to do here, but we need to talk about perception a little bit in this chapter so that, again, you can understand what patients and clients might be going through. First of all, if we have a receptor potential that's significant enough, we will send the signal up these ascending pathways. We sometimes call those tracts, okay? If it's a cranial nerve, it's going right to the brainstem, or it's going right to the area of the brain, and we'll talk about that, especially in special senses. When it comes to most of our somatic senses, or the visceral ones, that sort of a thing, you're integrating them oftentimes in places that do not reach conscious perception. That is to say, they are subliminal. If you've ever heard that phrase, subliminal, that means that there is a perception. It's just that it never reaches your conscious center. It requires a certain amount of intensity of signal, a signal that could probably be theoretically life-threatening or something you're particularly sensitive to. We call that phenomenon perceptual threshold. That is, and it's subjective. It's subjective to your experience, your emotional state, your previous previous life, previous events in that situation. It's called perceptual threshold. It's the level of stimulus necessary to become consciously aware of a given sensation. If you're not consciously aware of it, then we would call it subliminal. But if it's of enough intensity, or it's a type of stimulus to which you are particularly sensitive, when we think about people that have post-traumatic stress disorder, their perceptual thresholds for certain types of stimuli drop way down to where their hair trigger on certain things. But when the signal gets to the central nervous system, it is often sent to an area of the brain that is not the conscious center that will process that sensory information and then relay it to either other areas of the brain, like for instance your memory and affect, I forget the exact words there, your memory and your experience areas, your frontal cortex, and then on to your conscious center. How does that integration work? And we're just going to give you a sort of a generic mechanism. This is a big mech. Some areas receive some areas of the brain receive information directly, like for instance your olfactory pathway here, your olfactory tract goes directly to the olfactory bulb of the brain. That's in part because the olfactory sense is the oldest of the senses, we believe. It's the one that gave rise first, the original chemoreceptors, if you will. The other one that does not route to where we're going here are things that have to do with balance and equilibrium. For instance, if you slip on a banana peel, that's the best visual image I have, what you'll notice is that your arms will start pinwheeling before you even realize it. And the reason for that is that you have a need for speed. So what's happening is your equilibrium is going out and your body is compensating, because we don't need to involve the higher order neurons because it would take too much time. You would be on the ground before you had a chance to react to it. So when we have a need for speed, in the case of things like equilibrium, certain types of motor function, then we're going to integrate it either in the spine or in the cerebellum. And I'll get after that spinal one in just a second. So the two big ones, olfactory and equilibrium, both are integrated below the higher order neurons of the brain. Almost everything else routes through a structure we call the thalamus. The thalamus, not the hypothalamus. The hypothalamus is just right down here. That's a special monitoring set of neurons, by the way, which don't have a blood-brain barrier. But the thalamus is like your network operating center. Information comes in and it first goes to the thalamus, where it's then integrated to the appropriate brain region. So if you have the eye, it routes to the thalamus and then gets sent to the visual cortex in the back of your brain, ironically. If it's a somatic sense, like touch or so forth, it'll get routed through the thalamus into what we call the primary somatosensory cortex, which is kind of in the middle of the brain, around the side, and so forth and so forth. And different regions are in charge of processing sensory information, but it all routes through this thalamus. So most sensory pathways through the thalamus. Equilibrium is going to the cerebellum, and the olfactory is going to the olfactory. And there's a lot of interesting stuff to be had here. There's a lot of interesting psychophysiological phenomena, if I should say it. And this is a great example of this. Oliver Sacks, in fact, he was just on NPR, an old interview of his was on NPR the other day. Really, really interesting professor. He wrote this book, The Man Who Mistook His Wife for a Hat. Now, this isn't exactly sensory processing, but it's along the same lines. He took hold of his wife's head and tried to lift it off to put it on. He was perceiving his wife's head as a hat. Now, that's a visual processing issue. That's a visual processing. And Oliver Sacks himself had that problem. He once talked about being in a restaurant and he kept seeing this guy staring at him. He kept looking over and the guy was always looking at him every single time he was looking at it. And it wasn't until several minutes later and after much anxiety that he realized he was looking in a mirror. He didn't recognize himself. That's a sensory processing disorder, but I want to get after maybe one that's at a slightly lower level than this. So when we have sensory processing, we send it to the thalamus, which then routes it to the appropriate area of the brain, and then we coordinate and then we respond. If we have a problem in the thalamus, a given mode of stimulus, like for instance, you say auditory stimulus, might end up being sent to the visual center. Or if we, or other types of processing disorder. So we have to be able to process through the thalamus and send it to the right part of the brain in order to interpret mode. If it gets sent to the visual center, we will interpret it as a visual phenomenon. Location. How do we discriminate location? Well, we have a mechanism for that, and it involves this routing as well. Intensity, that's back to the idea of coding frequency. Remember how I made a big deal about relative refractory period in neurons? That's where we're going to see a bunch of this as well. That's part of this. Duration, how long the signal lasts. And we do get phenomena after images and things along those lines. But all of those things give us the flavor and the information we need to respond to it. So let's talk about modality, and I've mentioned this already. It's telling us what kind of a stimulus it is. How do we determine modality? What sensory neurons are activated, which receptors are activated, and importantly, which neurons receive that signal. So if we have a thalamic issue, maybe we perceive auditory as visual, or visual as auditory, or that sort of a thing. We have that adequate stimulus, and that certainly should play a role here. And then we have what is called labeled line coding. This is basically hard line coding to a given area of the brain. It starts with the afferent neuron, comes up the central nervous system, routes through the thalamus, and then gets sent to an area of neurons. We call it a nucleus or a cortex if we talk about the somatosensory cortex. And then that sensation is perceived. It's called labeled line coding. Okay? So if we have a misrouting here, we will have a different perception, even though the stimulus may be of a given mode. So if we're going to perceive touch, what we need is a mechanoreceptor activated through the thalamus going to the somatosensory cortex. If we are going to do proprioception, that's going to involve balance and equilibrium along with touch and stretch. Okay, so which neurons are activated? We're activating neurons in the muscles and that sort of a thing, but we're also activating neurons in the equilibrium area of the brain. And all of those guys are sending signals up into the higher order neurons to perceive where we are in 3 dimensions. Think of it this way. If you were to poke your finger right now with a needle, the pain would not be feeling inside your brain, even though you're perceiving it inside your brain. Nevertheless, you feel that pain external to your head. You feel it in your finger. That's another form of proprioception. Temperature, thermoreception, again, a type of mode triggered by a certain set of receptors that are most sensitive to temperature change in this case. And then we have nociception, what we call pain and itch. Again, free nerve endings are the most common here. We're going to break out pain and itch a little more carefully in this chapter. So we'll talk about that as a topic unto itself. We're getting close to the 30-minute mark, so I'm going to wrap up with maybe 1 or 2 more slides, and then we'll move on to part B. The next type of interpretation is location. And I'm actually going to use auditory stimulus for the primary example here, but let me show you where I'm going with this. When we have normal in-person class, what I like to do is have the students close their eyes, and they can easily discriminate when I am moving from left to right in the front of the classroom. And even some vertical displacement, if I stand up on a chair or something, they can locate that too. That has to do with the asymmetrical ear, by the way. But in any case, you can easily detect location left and right. And you can do it from behind too, but again, because of the shape of the ear, it's harder to discriminate vertical placement. That's what we mean by what we call population coding. That is to say, according to which receptive fields in a given population are activated. So multiple receptors functioning together. Now I alluded to population coding in that modality slide earlier when you looked at that gentleman balancing on that ball. He had multiple receptors functioning together. That included his equilibrium receptors as well as his proprioceptors in his muscles and mechanoreceptors in his feet and so forth. That's an example of population coding, which receptors are acting at a given time. We can also do this for intensity as well, but I don't want to go there just yet, but I don't want you to get confused, which I know is probably going to happen because this is complex stuff. Let me give you a simple example of population coding to look at in the ear. And again, this one's for intensity, not location, but I'll get to location in just a moment. If we have a very quiet sound, if it's very quiet, only a certain subset of the total population of neurons is going to fire. But as I raise my intensity of my voice, there's more energy in the sound waves and more receptors in that particular field are activated simultaneously. And again, population coding is important for intensity, don't get me wrong. Population coding can also distinguish pitch. When we hear different sounds, when the sound takes on a different timbre or a different pitch, different receptors are firing simultaneously. So we might have, if you listen to some music, you might hear the drums and you might hear the guitar, or you might, another time we might just have the horns, like for instance, a bunch of trumpets and saxophones playing. Population coding, which receptors are firing at which time. There's another phenomenon, and I want you to hold on to this. We'll come back to this in a little bit. Lateral inhibition. Lateral inhibition says that when you fire off one receptor field, and let's pretend that this word is a receptor field, this word is a receptor field, and this word is a receptor field. If we're firing off here, and even though it's intense enough that it may actually spill over onto these fields, this field will actually inhibit the fields around it. And that's for finer discrimination. But we'll get to that in a little bit. But hang on to that. Population coding first. Here's an example of population coding, though this is a temporal population coding. Here's our sound source, and our brain can easily interpret it as being left of center. Why? Because the population here sends signals before the population here. Even just a fraction of a second, the airwaves are slow enough that we can discriminate location even with objects relatively close to us. However, if we were to repeat this experiment underwater, there is no way you could discriminate where that sound is coming from, because sound waves in water travel so much faster, your brain can't process it quickly enough. Cannot process the left and right quickly enough. You may be able to discriminate intensity, but not likely. So in this case, it's population coding separated by time. So that's a good example of localization. Which receptors are activated and when. And you can try this experiment. Close your eyes, put your finger on your right hand index finger on the inside of your left arm and just move it up your forearm to your elbow. You can feel the different receptors being fired as it moves up towards your elbow. Population coding. So that does it for part A. Let's come back to lateral inhibition and finish up somatosensory perception in part B. Thanks for your time, and I'll talk to you soon.